Hey guys, so in this video I'm going to be going over mitosis and meiosis with y'all. So I know we kind of flew by with this, so I'm going to try to slow it down and really explain it to you guys, okay? So we are going to talk about mitosis first. Okay? And let's think about what we know about mitosis right now. So we know a couple of different words, vocabulary that's been tossed out, that's been explained, that is associated with mitosis. And one of those is going to be diploid. So what does diploid mean? Diploid means that they have, for every type of chromosome they have two of them. So say they have N number of chromosomes. So N chromosomes. Okay? Diploid would mean there's 2N. Okay? So there's two of each type of chromosome. But where does these two of each chromosome come from? Well, in humans, the two comes from one from, actually, I'm going to switch that to pink. So one comes from mom, and one comes from dad, right? So that is why we are diploid. So mitosis occurs in your somatic cells. Somatic cells are basically just all of your body cells, except for your sex cells. So most of the time, we are just going to be seeing mitosis in a person's body, right? Since meiosis is specifically for the production of gametes, or of your future babies. So let's go over the steps of mitosis that we need to know. So we're going to look at a real simplified version of an organism that only has two. So we're going to look at the simplified version with just two chromosomes, okay? So the blues mean they came from the dad. So I have one smallish one and one bigger one, okay? And then because we are diploid, we're going to have one from our mom as well, right? One bigger one and one smaller one. So what's going to happen first? So in order to split into two cells and go from one cell to two cells to an identical cell, what do we need to do? We need to replicate all of the material that we have in our cells. So when that happens, you get another little chromosome that just joins right here on the side, right? Just now it's got a buddy. So this term of this X shape right here, that's called a sister chromatid. So the sister chromatid is just the exact replicate of that chromosome. And it's the exact replicate because we are making two identical diploid cells with mitosis. So we want to double everything before it splits in half. So that way we start with this. So that way we end with the same amount of genetic material that we started with. So now that our chromosomes, which remember are only the one bar, and now that they're in the X, we have sister chromatids. So now that they're with their sister chromatids, they are going to line up at the plane of division. So we're just going to see them start to do their thing like this. So they line up at where their X, so their X in between the sister chromatids basically go onto this plane of division, And then when the cell splits, basically this sister chromatid is going to go to that cell, that sister chromatid is going to go to that cell, and so on for all of these chromosomes, right? Because if we have one sister chromatid going to each cell, that's going to be the same amount of DNA in each cell, not only to each other, but what we started with as well. So after this occurs, you're going to get the cell division split. So what does this look like? What do these new cells look like after the split occurs? Pause this video and draw it yourself, okay? And when you're done, unpause it. Okay, so hopefully you drew it out on what now these new daughter cells would look like. And they look like this. So they got the one long one, the one shorter one of the moms, and then you have the dads, which are the one shorter one and the one longer one. And you have that in both of them. Let's make these a little bit longer to size. Okay. So does that make sense? So that is mitosis. And basically what mitosis did is it went from a diploid parent cell to two identical diploid daughter cells. So I want you guys to make sure that it's clear that when we talk about parent cell and daughter cells, we're not talking about our mom and dads. Like we're not talking about like mom and baby organisms or nothing like that. We're talking about the parent cell as the original cell. And we're talking about the daughter cells as the new cells. Okay? So, now that we have mitosis, let's go and talk about meiosis. So meiosis. So meiosis is the process where we go from a diploid parent cell to 4 different nope, I don't want that. No. Okay, to 4 to 4 haploid daughter cells. Okay? So we're going to go through what exactly this means, the important phases in meiosis that you guys need to know, and how this differs from mitosis. So first of all, meiosis only occurs in sex cells. So what are sex cells? Sex cells are your ovaries and your testes, right? Things that are going to basically produce an offspring, okay? So meiosis is part of a process called gametogenesis. Okay, and we're going to go through that as well. So meiosis, what happens here is a little bit different than mitosis. So we have our big chromosome, we have our big dad chromosome, our small dad chromosome, big mom chromosome, and small mom chromosome, right? So the first thing that's going to happen is that they need to replicate. So that's the same thing as mitosis. So they're going to get that little sister chromatid joining on them, right? Sister chromatid. Okay, so now everyone is happy with their sister chromatids, their exact replicate of the chromosome. So now we're going to set up the plane of division, but this is where it's different from mitosis. So mitosis, remember, they lined up based off of their X's, okay? So they all lined up, their X's were basically in the center, right, of the plane of the division. So can you see that? But this is not what's going to happen in meiosis, at least not for meiosis 1. So right now what we're observing when we start with a diploid cell is meiosis 1, okay? So for meiosis 1, how they're going to line up is a little bit different. So your sister chromatids are going to go like this near the plane of the division, right? So let's make these ones a little bit bigger. But here, now instead of, so these ones will also line up near the plane of the division, but now part of that chromosome is going to cross over. Part of the dad chromosome is going to cross over with the mom chromosome. So let's cross that part over too. Okay, so this crossing over, okay, so crossing over is how we get genetic diversity. It basically recombines your mom's DNA and your dad's DNA to make a chromosome that has never been formed before. Okay, so it's a unique chromosome. So this process is called crossing over or recombination. Those terms mean the same thing, and they make unique chromosomes. Okay? So now, when these get pulled apart, because remember, in mitosis, after they're on the plane of division, they get pulled apart. So that's going to go to that cell, that's going to go to that cell, that to that one, that to that one. So I want you to pause this video again, and I want you to try to draw what these new cells would look like once this separation occurs, okay? Okay, so once this separation occurs, this is what these cells are going to look like. So let's look at the mom, the cell that's going to have the mom's chromosomes for the most part. So we have our big one, and then instead of drawing a big one that fully completes it, I'm going to put a little bit of blue on the top. Okay? And I'm going to do the same for the small one. Now, why am I going to do this? Well, when this crossing over occurred, what happened was the DNA swapped between the two. So crossing over is like swapping DNA. Okay? So on this side, on the dad's side over here, we have our big chromosome with its sister chromatid, but I'm not drawing all of it because there's a little pink piece here from the mom, right? And then the same thing with the smaller one. So now these are unique chromosomes. So these 2 chromosomes, or should I say these 4 chromosomes, these are unique, okay? These chromosomes did not exist beforehand, and now they do. So basically, they're going to line up on the plane of division, just like with mitosis. So this looks about center, so it's going to line up here like that, and it's going to line up there as well. So it's going to line up like mitosis now, okay? So this is now considered meiosis. I spelled that wrong. I totally butchered that. This is called meiosis 2. So meiosis 1 is where when they line up on the separation plate, on the plane of division, there we go. I forgot what it was called for a second. Sorry. So when they line up on the plane of division for meiosis 1, you get the homologous pairs joining, okay? And just to clarify the word homologous pairs, that is the easiest way to compare this is to shoes. So you have a pair of shoes, right? Your left and your right shoe are slightly different, right? But they are, for the most part, the same. So homologous pairs are the same chromosome, but they contain different versions of genes. Okay? So that's like if your mom has blonde hair and your dad has black hair, then you're going to have, in your cells, you're going to have the alleles for both blonde and black, okay? Different versions of a gene, which the gene is, it would code for hair color. So let's go back to what's going to happen next in meiosis 2. So I said they're going to line up on the plate, on this plane of division, which I already did that there, so I'm just going to erase these real quick. And we're going to move on to what happens next. So what happens next is these cells are going to separate, right? Now is a good time to pause this video and draw this yourself, okay? What are these 4 haploid cells going to look like now, okay? So you can go ahead and pause it. Okay, so let's look to see what these ended up looking like. So we're going to have one of the mom's big original chromosomes, and then let's say we have one of the smaller ones, but this is the recombinant chromosome, okay? And then we're going to have the mom's recombinant chromosome, the big recombinant chromosome, with the smaller, just regular mom chromosome. And here we'll have the dad recombinant chromosome, big one, and the small non-recombinant one. Or, you know what? Let's say in this cell we have both of the recombinant ones. So we also have the small recombinant chromosome. And then here we have the dad big chromosome and then dad's small chromosome. So why did I switch up the fact that in each one of these 3 there's one of the original ones and one of the recombinant? Whereas this one, I was going to put the one of the originals with one of the recombinants, but instead I decided to put the two recombinants together and the two originals together. How could I have that decision to choose either or? I am so sorry for my dog barking. Him and my cat are playing right now. Titan! Shh! So, why did I choose to do that? How did I have that choice? Well, that's where the concept of independent assortment comes in. So independent assortment is basically the fact that all of these chromosomes in here, they didn't specifically have, so this one didn't specifically have to go into this cell, and that one specifically didn't have to go into this cell. It could have been vice versa. This chromosome could have went into this cell, and that one could have went into that cell. So the fact that they have the choice, and it's by not so by choice, but it's by random chance as to which one they go into, that is another component that creates genetic diversity. So in meiosis, when we're thinking about the factors that now make these 4 haploid cells unique, well, first of all, the things that make them unique, 1, they're haploid. So they have half the number of chromosomes. That looks weird. Of chromosomes than the parent cell. Okay? 2 is, well, they underwent recombination. They swapped their DNA. And the third thing that causes us to all be unique and have diversity is this independent assortment. So I hope that cleared things up with mitosis and meiosis. Before this video ends, I just want to briefly go over gametogenesis. So gametogenesis is the formation of gametes. So, gametogenesis, it follows meiosis, okay? So, it's going from a diploid parent cell to 4 haploid daughter cells that are genetically different. Okay? So, for the males, so the special term for Titan, we're not doing this. Okay, I'm going to pause this video, put him in his cage, and then I will continue.